I'm excited to talk to you about my recent research as a NASA astrobiology postdoc in the laboratory of Victoria Orphan at Caltech. And at the end, I'll also be discussing briefly a few new projects I'm starting here at Georgia Tech. So here's a brief outline for what I'll be discussing today. First of all, I'm going to talk about why do we care about methane and why do we care about metals. And what do we already know, and what is new that I'm going to be sharing with you. And finally, what am I going to be doing next? So why do we care about methane? Well, methane is a very potent greenhouse gas. It's actually the second most abundant greenhouse gas in the atmosphere. And after about a decade hiatus in the 90s, methane concentrations are now rising again. We don't exactly understand the cause for this, but we think that it probably has to do with high latitude warming and release of methane from permafrost and wetlands. So microbes are really at the center of methane sources today. Here you see that about 2/3 of all methane sources are microbial in origin. So where exactly are the microbes that are driving the methane cycle on the tree of life? So here you can see the 3 domains of life, and the microbes that are making methane are in the archaea. They are in the Euryarchaeota. And the microbes that are consuming methane, methanotrophs, are more phylogenetically diverse. They occur both in the bacterial domain and in the archaea domain that I'll be talking about today. These include the gamma proteobacteria, the alpha proteobacteria, and the Microbia in the bacteria, as well as a newly discovered phylum called MC10. So these are all aerobic methanotrophs. And over here, phylogenetically related to Methanosarcinales, are the anaerobic methanotrophs I'll be talking about more today. And I'm particularly interested in metalloenzymes that are involved in microbial methane cycling. So here on the left, you see a schematic for methanogenesis coming from acetate, CO2 and H2, or methyl substrates. And you can see that there are abundant metalloenzymes involved in these processes. Most obviously, there is a lot of iron involved in these enzymes. But other cofactors include cobalt, nickel, zinc, molybdenum, and copper. And here on the right, I have outlined the 2 major pathways for aerobic methanotrophy. There is the high copper pathway and the low copper pathway. So this aerobic methanotrophy is really, copper is really important for these, for controlling which pathway is used. When copper is low, iron is utilized in the soluble methane monooxygenase instead of the particulate monooxygenase that is a copper protein. Now I'm really going to be focusing on anaerobic methanotrophy today, particularly reverse methanogenesis used by anaerobic methanotrophic archaea. But there are some other really interesting results I just wanted to highlight before I get going, including a recent discovery that methanol dehydrogenase can actually be substituted by lanthanides for growth. And lanthanides are used in place of calcium in this key enzyme. And this is really exciting to me. This just came out in 2013 because this is the first time that lanthanides have ever been shown to be involved in biology that I know of. And it's these kind of results that get me really excited about new discoveries in microbial methane cycling. I also just wanted to highlight that there is another microbe that was recently discovered called Methalomoleculus oxyphera, which combines denitrification with aerobic methanotropy. So it's actually taking nitric oxide and dismutasing it and turning it and producing intercellular oxygen, which is then used for aerobic methanotropy. And these cells are very interesting polygonal shape, and it's not quite understood exactly why. So as I said before, I'm going to be primarily focusing on anaerobic oxidation of methane today, abbreviated AOM. And this is performed by symbiotic associations of anaerobic methanotrophic uriarchaeota, or ANME, and sulfate-reducing bacteria, or SRB. And throughout the talk, I'm going to be using red for ANME and green for SRB. And these organisms occur primarily in marine sediments, where there is abundant sulfate. And the net reaction here is methane and sulfate going to bicarbonate and sulfide. And there is an unknown intermediate that is presumably transferred between these two organisms. Recently, just in the last year, it was suggested that zero-valent sulfur could be the intermediate between these two. This is still, however, a topic of very active research. And the reason that these organisms are so important is that 80 to 90% of the methane that is produced in marine sediments is consumed by these symbiotic associations. And so here, as I showed before, the ocean here in this tiny teal sliver is only 3% of the global methane sources. And the reason for this is that so much of the methane that's produced in these marine sediments is being trapped on the seafloor by these organisms. So there are no pure cultures currently available of these symbionts. They have very slow doubling times, 4 to 6 months, and so it's quite difficult to work with these in the lab. And we have to use other techniques such as metagenomics and metaproteomics to try to understand the metabolisms that these organisms are performing. So a bit more about the geochemistry of these seep systems. So here you have the sediment-water interface, and here you have oxygen, and oxygen is very rapidly depleted in these methane seep ecosystems. And sulfate is also depleted, as you can see, and produced is sulfide, shown here. This is being produced by the sulfate-reducing bacteria. And sulfate reduction and anaerobic oxidation of methane are coupled on a close to one-to-one stoichiometry here at the sulfate-methane transition zone. So here are just a few pictures of what these methane cold seep ecosystems look like. Again, they are cold. They are 4 to 10 degrees. This is on the continental slopes, so this is in the deep sea. These are cold ecosystems. And as I mentioned before, these organisms are actually producing bicarbonate, which combines with the calcium in seawater to produce these large calcium carbonate rock deposits. And these actually build up to very large scales, really mountains on the seafloor. And they harbor really interesting macrofauna and microbial communities. So my particular focus, again, is trace metal utilization by these consortia. And I want to highlight a few of the enzymes that are utilizing metals in these pathways. Sulfate reduction performed by the sulfate-reducing bacteria is requiring zinc, cobalt, and iron. And anaerobic methanotrophy using reverse methanogenesis, shown here, is using presumably nickel, cobalt, molybdenum, or maybe tungsten, as well as iron. However, we don't really know what metals are involved because these organisms have never been cultured before. So again, we have to rely on these omic techniques to try to resolve what enzymes are being utilized. And recently, there was a discovery by the Orphan Lab, my colleague Ann Deakus, that these ANME can also perform nitrogen fixation. And I'm going to discuss in a moment what cofactor we think is involved in this process, whether it's the molybdenum form of nitrogenase, the vanadium form, or the iron form. And the reason that I'm particularly focused on looking at metal availability in these systems is that we think that they may be metal limited because the sulfide that is produced by sulfate reduction is known to scavenge metals out of solution by forming metal sulfides. And you can see these metal sulfides in these dark pyritic sediments here. So all of this dark clay is filled with metal-rich sulfide, pyrite. And so first I'm going to be discussing a bit of what we know about the enzymes in this system from previous studies, particularly the nickel-containing cofactor methyl coenzyme M reductase, which is particularly important for anaerobic methanotropy because it catalyzes the very first step. So there is abundant evidence for use of Ni-MCR by ANME archaea. This comes in the form of metagenomics. Here is a phylogeny of MCRA genes from metagenomes from methane seep ecosystems from all around the world. And also, we can see that this gene is actually expressed in these aggregates shown here. This is fluorescent immunolocalization of the MCR gene in the MCR protein, in archaea, lighting up here. These are the sulfate-reducing bacteria that are not lighting up. This has also been shown with immunogold localization by other groups. And actually, 10 years ago now, this cofactor was isolated from microbial mats in the Black Sea and shown to contain nickel. So it's quite well known that these organisms require nickel in fairly high quantities. 7 to 10% actually of their total proteins are this MCR that contains this cofactor F430 with the nickel inside of it. And we've actually been going through the metagenomes and looking for further evidence of exactly how these organisms are obtaining and using nickel. And we found 22 sequences in the metagenomes with closest identity to uncharacterized metal chelotases in methanogens. And looking through the literature, it suggested that actually these uncharacterized chelotases are used in cofactor F430 biosynthesis. However, this whole pathway, the biochemistry of it, has actually not been worked out yet. But this is further evidence from our metagenomes that these organisms are indeed making cofactor F430. And we've also been looking at transporters for nickel in these metagenomes. And this is the most common nickel transporter in bacteria and archaea. It's Ni-MNOQ. It's an ABC transporter. And we actually found this in our metagenomes from 3 different systems that I'll talk more about in a moment. And we found this in both the ANME and in the SRB. So further evidence of the importance of nickel and that these organisms can acquire nickel from pore waters. So just a bit more to set up the story about what we know. Now I'm going to turn to nitrogenase and consider its metal cofactor. So the Orphan Lab has been studying nitrogenase for quite some time in ANME. And Ann Deakus in Science in 2009 showed, using a combination of fluorescence in situ hybridization and nanosims, that these archaea are incorporating labeled nitrogen into their biomass. And further study of the phylogeny of the NIFD gene that encodes nitrogenase show that in the methane seep nifty clade, it is lying between the alternative nitrogenases. It's cut off here, but this is the vanadium iron and the iron nitrogenases. And characterized molybdenum nitrogenases. So it was unknown whether these organisms were using the alternative nitrogenase or the more commonly observed molybdenum form of nitrogenase. And my colleague Sean McGlynn then has delved further into this phylogeny, and he has resolved now that it is likely the molybdenum form of nitrogenase that is contained in these ANME metagenomes. So here you see in red, this is the uncultured archaea, and its nifty gene is lying in between these characterized forms of the molybdenum form of nitrogenase, whereas down here is the iron form and here the vanadium form. So when I came into the lab, my question, coming as really a geochemist, was, well, what are the concentrations of trace metal micronutrients in these very sulfidic seep sediments, where presumably a lot of metals, micronutrients, are being trapped as sulfides? And can the microbes actually access these metals bound to sulfides, or could they be actually metal limited? So to do this, I had the good fortune of being on a cruise with the Orphan Lab on the RV Atlantis using the Jason ROV in 2011, and we looked at seeps at Hydrate Ridge off the coast of Oregon, fairly close to shore, at about 600 meters depth. This is showing a map of the system. You can see these enormous mountains. These are the carbonate formations that are really driven by the chemistry that these, the byproducts of these microorganisms. So there are a few different major types of habitats in these methane seep ecosystems. There are calyptogena clam fields, where these clams actually contain sulfide oxidizing endosymbionts. And there are abundant Biogeochemical microbial mats covering the seafloor that are also oxidizing sulfide. So obviously this is a very, very sulfidic system, and these organisms are really, on the surface, are really living off of the sulfide that's being produced by the sulfate-reducing bacteria. So we collected samples, as you can see, these push cores from all three of these types of ecosystems. And we also explored some very small-scale geochemical heterogeneity between adjacent cores. So here you see this mat, which is interestingly, you can kind of see it here. It's a quite bright pink color coming from the cytochromes that are contained in these Biogeochemical mats. And right next door, we have another mat that was a white mat. And we looked at two of these cores, quite adjacent to each other, only 1 meter away. And this core actually had lower sulfide and lower, these are the ANME SRB aggregates here. And I measured by ICP-MS the metal content of both the sediment and the pore water. And then I did the same for this pink mat. And you can see sulfide concentrations were quite a bit higher, as were the ANME SRB aggregate counts. And the metal concentrations for both the sediment and the pore water were generally depleted compared to the adjacent core. So given that there's more sulfide and more aggregates here, we thought perhaps that this metal depletion was actually a result of the microbial communities taking up metal from the pore water. And so we further explored this, looking at the dissolved metal concentrations in the pore waters from each of these habitats. And so here we used, this was 43 push cores from the mat, 23 from the clam, and then 18 from the periphery. And so here you can see the general trend is that cobalt was actually our scarcest trace metal in these systems, followed by nickel, molybdenum, and iron. Interestingly, molybdenum concentrations were quite high, which may explain why we have the molybdenum form of nitrogenase being expressed. And here in these gray boxes, I've plotted the optimal concentrations that I collected from literature surveys for growth of methanogens, which again are related to the ANME archaea that we were studying. This was as good as we could get because we don't have studies yet of pure cultures of the ANME. And so you can actually see that furthest from the optimal concentrations is the cobalt in the pore waters. So this led us to think perhaps cobalt could be limiting growth in these systems since it's so far under the optimal concentrations. So where do we see cobalt use in enzymes in these AOM consortia? Well, cobalt is actually really important for both anaerobic methanotropy and sulfate reduction. It's present in the MTR enzyme, which is the second enzyme in reverse methanogenesis. And it's also present in the acetyl-CoA pathway, the Wood-Youngdahl pathway, for conversion of this carbon from methane into cellular biomass. And some studies also suggest that it's used in the first enzyme in sulfate reduction as well. And there is evidence in the literature to suggest that methanogens and perhaps ANME archaea could be limited by cobalt scarcity. Here is a study of Methanosarcina barkeri in sulfitic media showing that you see quite a difference in the amount of methanol consumed between 400 micromolar cobalt and no added cobalt, somewhere under 2 micromolar cobalt. And this is around where our system is lying, somewhere in here. So we were wondering whether it could be, in fact, limiting. So there's that question. And we did an experiment in the lab using Hydrate Ridge sediment. Shown here, we used actually sediment that had the lowest cobalt concentrations of all the pores that we measured. Did this all anaerobically. And we actually, however, found no difference in the amount of sulfide produced, which is a proxy for the amount of AOM happening, between the no added cobalt condition and the 2 micromolar cobalt addition. As you can see, then the kill control did not produce sulfide. And this was a 70-day incubation. So this leads us to wonder, how do these consortia then acquire cobalt at very low concentrations, at nanomolar levels? And also, could cobalt be shared between members of the AOM consortia, given that we don't fully understand the intermediates involved in these symbiosis? So here I just want to explain in a bit more detail the methods that were being used, which I've mentioned a bit before. We are using metagenomics and metaproteomics. And so I'm sure many of you in the room, I know, are more familiar with me than with metagenomics. But this technique of actually separating the aggregates from the sediment in these methane seep cores was developed by the Orphan Lab, and it's called magnetofish. And what Pernthaler et al. Did was basically to use species-specific DNA probes attached to magnetic beads to pull out the ANME archaea and the SRB that they are in consortium with. So here you can see examples of that. And then to extract that DNA only and to sequence only that DNA so that you're getting basically from the bulk sediment, you're isolating just the DNA from the actual AOM consortia. Of course, there are many other microbes in this sediment, and we were particularly interested in these consortia, so we have to separate these out from the bulk sediment. And so then we take these metagenomes from just the isolated organisms, and we use these to make a database that we can search when we run the bulk sediment metaproteome. We don't have a good way of just isolating these organisms for protein work, so we have to use bulk sediment and extract proteins from that, digest them, and run them. This is in the Hettich lab at Oak Ridge National Laboratory. Again, but then we can basically be sure that the proteins that we're looking at that are expressed in by these consortia are coming actually from the ANME SRB themselves, as opposed to something else in the sediment that's not our consortia of interest. And so the Orphan Lab has been sequencing these metaproteomes from Hydrate Ridge, Eel River Basin, and Santa Monica Basin up and down the West Coast offshore. And we have been using these then to get at various questions, including the one I posed before, which is how do AOM acquire cobalt and other trace nutrients at nanomolar levels? And so here I show phylogeny of cobalt transporters. All the black is for cultured methanogens, and then the red is where we found it in ANME. So these are the bead capture metagenomes, and this is also a bulk metagenome. But we can be sure that this is actually, these two are coming from the ANME. And this is for the CBIM transporter. This is the most common type of cobalt transporter in archaea and bacteria. And it's actually homologous to the Ni-MCR and OQ that I showed previously. So we think that this is probably being used by these organisms to acquire cobalt at nanomolar levels. So then we looked further into the cobalt utilization by delving into vitamin B12 pathways. So vitamin B12 is the most common cobalt cofactor in biology, and some microbes can synthesize vitamin B12 while others cannot. So we were wondering maybe if cobalt or vitamin B12 could be shared between members of these consortia, perhaps as the intermediate or one of the intermediates involved in this symbiosis. So we looked into the vitamin B12 biosynthesis pathway. This has 15 to 20 steps, each performed by a different protein. And in the metagenomes, we found that we could identify most of the genes for the B12 biosynthesis pathway in both the ANME and the SRB. So the boxes here represent genes that we were able to find, and then the asterisk represents where we actually found a protein that was expressed in a metaproteome. So in this case, we only found the CBIA, which is actually a key protein involved in this biosynthesis pathway, in the SRB. And here you can see that it's blasting to our bead capture contigs. So we could be sure that this was actually coming from the SRB partner as opposed to another sulfate-reducing bacterium in the sediment. So luckily, the Orphan Lab had also been doing some very long-term 6-year incubations with just 2 transfers where they had added vitamin B12 to the sediment, and they wanted to see whether if we added vitamin B12, could we actually observe that the 2 organisms, the ANME and the SRB, would separate, basically. So if this were an intermediate and we gave it to them, they wouldn't need to be in symbiosis anymore. However, what we observed when we did fluorescence in situ hybridization, with the red being the archaea, the green being the bacteria, and then the blue just being DAPI, is that we still found primarily bundles of these archaea and SRB. So this suggests perhaps then that vitamin B12, although it is probably very important for these consortia, their metabolisms, it's probably unlikely to be involved in the symbiosis. So now I'm going to move on to perhaps our most exciting result from this study, which was looking at the question of this, what is the cofactor in this last enzyme in reverse methanogenesis, taking formal methanofuran to CO2. This is the first enzyme then in methanogenesis, the last in reverse methanogenesis. And we know from the literature that this enzyme can either be molybdenum or vanadium. In the molybdenum form, it's called molybdopterin, it's the cofactor, and in the tungsten form, it's tungstopterin, so molybdenum or tungsten. When we looked at the availability of molybdenum and tungsten in these porewaters, we found that molybdenum was much higher in abundance than tungsten. Here's tungsten in the pink and molybdenum here in the purple, and molybdenum is orders of magnitude more abundant, about 3 orders of magnitude more abundant than tungsten. So we expected then to find the molybdenum form because the molybdenum concentrations were higher. We already knew that the molybdenum form of nitrogenase is used by the ANME archaea, and also only hypothermophilic, only thermophilic and often hypothermophilic methanogens are known to use this tungsten form of this enzyme. However, when we created a phylogeny of these formal methanofuran dehydrogenase genes, again, black is just showing all the methanogens we could find in the NCBI database, and we then added our metagenomic sequences to this, and in red you see those sequences. So we can also add to our phylogeny then the characterized methanogen enzymes. So this in yellow here and here, these enzymes have actually been purified and their metal content characterized. And so we know then that this contains tungsten, tungstopterin, and this contains molybdopterin. So we can then assign each of these clusters, this being the molybdenum cluster, or FMD, and this being the tungsten cluster, or FWD. And this is the B subunit, which is the catalytic subunit. And so clearly then, and this is actually, we don't have any yellow, we don't have any characterized enzymes, so we don't know exactly what the cofactor is, or even what these enzymes are doing. So presumably they're doing the same reaction, but we really don't know yet. So then in the asterisks again is where we actually see this gene being expressed in the metaproteomes. And again, we can be sure that this is coming from the ANME SRB consortia as opposed to the bulk sediment. And we find this in both the Hydrate Ridge and the Santa Monica methane seep metaproteomes. And so this is lying in our tungsten cluster, and so we can be fairly certain then that these ANME are actually expressing what appears to be highly similar to the tungsten form of the enzyme. And further evidence of this comes from then the gene order. There's actually a different order of the operons for the molybdenum form and the tungsten form. And when we look at where this came from, this is actually blasting closest to a phosmid in the Eel River Basin from Hallam 2004's metagenome. And when you look at the phosmid there, actually the order then that we see is this order, this DBAC. So that's further evidence then that it is the tungsten form that's being expressed in the metaproteome. So again, this is just showing our table of these two hits in the metaproteome that seem to be the tungsten form. So this we found to be pretty exciting. And actually, it has been observed once before in 2012. I dug into the supplemental material from this other methane seep off of Norway, and they didn't actually mention this in the paper. In fact, in the paper, they labeled it as the molybdenum form. But in fact, it also is blessed and closest to the exact same phosmid from the Halan metagenome that we found in our Hydrate Ridge in Santa Monica. So it seems that this is actually, it is being expressed in at least 3 methane seeps globally distributed. So it seems so far that it could be important in, globally in anaerobic oxidation of methane coupled to sulfate reduction. So as I mentioned earlier, we really find known tungsten users using microbes to be, they're only in the bacteria and the archaea as far as I know. And they're primarily either mesophiles or thermophiles or even hypothermophiles. So I put the temperatures here of the organisms with characterized tungsten-containing enzymes. And Michael Adams at UGA has actually done a lot of work on this Pyrococcus species that was isolated from a hydrothermal vent. So it's commonly, in the literature, tungsten use is associated with ultra-high temperatures. And so this study is kind of changing that paradigm a bit by showing that the ANME growing at 10 degrees in these methane seep cold seeps seems to be expressing a putative tungsten enzyme. So this work that I've just told you about has been, is now accepted in environmental microbiology. It should be coming out anytime now. And in summary, I just wanted to leave you with my takeaway message. You remember one thing from this talk. We seem to find tungsten in psychrophiles as well as hypothermophiles. So then what is next on the agenda? Well, now, actually, throughout my postdoc, one of my major aims was to actually map the elemental distribution in these AOM consortia. And I tried to do this with nanosims, and I actually had quite a hard time getting the detection limits low enough in order to do this mapping of the actual trace elements. So now I'm using the advanced photon source at Argonne National Laboratory, specifically the Beamline 21 IBD, which is a hard X-ray fluorescence nanoprobe that actually has cryogenic capabilities. And we're using this to do XRF mapping and microspectroscopy in our AOM consortia. And I just wanted to highlight a few of the recent really exciting results that have been shown, and then just one slide of what our preliminary results look like. So this is quantitative 3D trace metal imaging of a centric diatom that was published 2010. And what I find really exciting about this is that you actually see manganese and iron in the silica frustule in this centric diatom. And these results were very controversial at the time. This had never been found before, but actually Ellery Engel, my colleague in EAS, has just verified this in Nature just this year, that in fact, yes, there is iron incorporated into the frustrules of these diatoms. So this is one exciting result. Another exciting result has come from mapping the trace element distribution in Trichodesmium, which is a major marine nitrogen-fixing cyanobacterium. And what this study found was actually that vanadium was the most abundant trace element in trichodesmium. Usually either iron or zinc is the most abundant metal. In this case, it was vanadium. It's not exactly sure why vanadium is most abundant. It's thought perhaps that these trichodesmium are phosphorus limited, and the phosphate and vanadate transporters look very similar, so perhaps they're taking in vanadate as they're trying to get phosphate. And so, as you can see here, the great advantage of micro-XRF imaging is that you basically have access to most of the entire periodic table. Unfortunately, not molybdenum, but most of the other trace elements. And so we've started to do this mapping, and here are just very preliminary results. Here in the left corner, this is one of our aggregates. This is at low resolution because actually our run was canceled, so we had to. We only had 3 hours to run. So with 3 days to run later this month, hopefully this will look much better. But what you can see then is clearly sulfur, which we know is actually elevated in the ANME. So likely this is the ANME we're seeing here. And as well, we're seeing this ring or crust around the aggregate. And this is very interesting because we think perhaps that the consortia are basically binding clays on their on the outside of the aggregates. And it's not exactly sure why this is happening. There's some crusted aggregates, there's some uncrusted, but it's possible that this is a mechanism for them to acquire micronutrients by actually gluing them to the outside of the aggregates. And I also just wanted to highlight a few of the new projects that I'm involved with at Georgia Tech, and many of the people in this room are on this slide because I've had great initial collaborations with many people. And so we're continuing the anaerobic oxidation of methane work. This is with Tom DiCristina, as well as Frank Stewart. With Joel Koska, I'm looking at interactions between methane, metals, and nitrogen cycling in peatlands for a DOE proposal. I'm also very interested in expanding out to nitrous oxide. It's a particular interest of mine in oxygen minimum zones. And so this work in oxygen minimum zones is with Frank Stewart, and I had the great opportunity of having him invite me on his cruise last June, and so we've collected some primary data for this. And I'm also working on a new instrumentation center here for ICP-MS. We don't currently have a collision cell ICP-MS that we can use for ultra-low detection limit work on trace metals. And so we're putting in an MRI, and many people in this room are users on that. And hopefully we get that funded, and so we can have a great new instrumentation center here. So thank you all for coming today, and I just want to highlight this work was done in Victoria Orphans Lab, and it was funded by a NASA astrobiology postdoctoral program. And I want to put out a plug also for the 2014 Southeastern Biogeochemistry Symposium. This is primarily student-organized symposium, and some of the students I think are in the room that are organizing this. Jordan and Aaron and Keaton, I don't know if he's here. So this is April 5th through 6th, 2014, Georgia Tech. And really anybody working on any aspect of biology and geochemistry or any combination thereof is welcome to come. This is all going to be student and postdoc oral and poster presentations, and our keynote speaker is the author of Biogeochemistry, now in its 3rd edition, Bill Schlesinger. And I just want to say I am looking for students. So if you know of any great undergrad or level students, please send them my way. So thank you.
We have time for a few questions. Joel.
Jim, why would ANME fix nitrogen when they are washed in ammonia?
The Yes, I think that's, well, when Anne and others in the orphan lab were measuring the ammonium profiles, they're quite variable. So they're perhaps in these micro niches, they're not always awash in ammonium. And also, since the ANME are on the inside mostly of these aggregates, you know, it's possible that, you know, not as much ammonium is accessible to them. But it's a really, I mean, I think it's a hard question. But I think it's a question that's, you know, from the studies we've been seeing coming out, it's an open question as to when do you see nitrogen fixation in marine sediments, even when you measure, you know, high micromolar levels of ammonium, you still see nitrogen fixation. So it's definitely being observed elsewhere.
Maybe one more? Do we have one more question?
Sure. You told us to quite a big microphone. You told us to, when we leave here, to remember that we can find tungsten in the psychrophile.
Yes.
But I don't know why I need to remember that. Can you tell me why that's important?
Well, it's suggesting that, you know, there's more kind of, you know, there's greater diversity in terms of, you know, metal use than we previously thought. And it's, yeah.
Is the claim that we used to associate tungsten with just thermophiles and hyperthermophiles?
Right, that's...
And is there something about the valence of tungsten that is preferred at high temperatures?
It's actually a matter of sulfide is more reactive with molybdenum at these hot temperatures. So you get these huge molybdenum sulfide deposits, and tungsten is actually elevated. But in our system, we see higher levels of molybdenum than tungsten, so we were expecting molybdenum. However, it's presumably a matter of speciation then in our system that for some reason tungsten is more bioavailable than we thought. It's really, I don't really know exactly why is the answer to your question, but I think it is interesting, and it is you know, a key enzyme in this process, which seems to be globally distributed. So I would argue that it is important to understand, you know, the mechanisms.
Okay. Oh, thanks.
All right, we have a class coming in next, so we need to move out. Thank you, Jim.